Okay then, you can get started. Sorry, who was speaking? Let me remind you.
Yeah, Suraj. Yeah.
Okay, Suraj from Genomics One. Please go ahead.
Yeah, sure. Determining the order of DNA building blocks, that is nucleotides in an individual genetic code, is called DNA sequencing. There's mainly of two types: whole exome sequencing and whole genome sequencing, which is automated with the latest technology called the next generation sequencing, which is much faster and more efficient sequencing technique. Exome sequencing is basically exome is composed of all the exons within the genome, and the sequences which are when transcribed remain within the same mature RNA after introns are removed by RNA splicing. This includes untranslated regions of mRNA and coding sequences, and the method of this sequencing of these exomes is called exome sequencing. This method allows variations in the protein coding regions. I guess my voice is echoing from others' mic.
Yeah, I think everybody should get mute, please.
Yeah, this method allows variations in the protein coding region of any gene to be identified rather than in only few select genes, because most known mutations that cause diseases occur in exons, and whole genome sequencing is a thought to be efficient method to identify possible disease causing mutations. The genetic disorder of our interest is DMD, that is Duchenne's muscular dystrophy, which is one of the most common type of muscular dystrophy which are inherited in the X-linked inheritance pattern. A germline mutation in the DMD gene, which is also the largest gene in human genome, causes the formation of non-functional dystrophin, which is a protein which imposes a high chances in causing progressive muscular degeneration and weakness. Muscular dystrophies are genotypically and phenotypically heterozygous. Exome sequencing here has been described as one of the most efficient and usually fast and accurate means of identification of the genes and mutations implicated in DMD when compared to Sanger method. The main objective is to carry out exome sequencing analysis for DMD, and then we aim to identify genes and mutations implicated in DMD and possibly detect novel genetic causes involved, and to identify variants in known and novel diseases genes. The methodology used is based on exome sequencing for identifying causative variants in disease diagnosing pipeline, which is based on the web-based Galaxy EU by Wolfgang 2021. And yeah.
Okay, so we first get the data from the databases and prepare them in Galaxy. We perform the quality control of the datasets and mapping them with the reference genome. Then we filter and remove the duplicates and continue the analysis with variant calling and variant annotation. After processing with inheritance processing with inheritance pattern tools, we finally visualize the VCF file using JBrowse or IGV. The exome sequencing and variant analysis will enable us to identify base and amino acid changes and the class of mutations such as missense, nonsense, and silent mutations which are observed in the dataset. Additionally, we will be able to detect the type of variants like SNP, MMP, indels, and some of which may cause the loss of function or likely pathogenic variants producing a truncated dystrophin protein causing DMD. The findings of our research pipeline may define new biomarkers and help us in contributing to better diagnostics of DMD and be beneficial for the future treatments. Thank you all so much for listening to us. If you have any questions, we will be happy to answer them.
First of all, the second speaker, you did not introduce yourself. I did not get to see the speaker. Second speaker, can you just introduce yourself?
Oh, sure. This is Negar Khadili.
Okay. Yeah, questions from the audience, listening, from the press, gentlemen of the press. Yeah, asking a question and answer if you ask in chat if you're interested. Okay, so from me, I have a question for you guys. Do you guys think you are, okay, someone asked, what are the variant inheritance tools you would use? I don't know. I think it's asking about the tools you will use for variant calling.
Yeah, sure. The variant inheritance tools which we are going to use is from Gemini, Gemini
Okay.
has a class of tools under which we are going to focus on the Gemini inheritance tool. That is the name of the tool, which basically gives out you can basically sequence based on the type of inheritance you go ahead with. It can be autosomal dominant or autosomal recessive or even allosomal dominant and recessive. So the tool which we are going to use is Gemini inheritance tool.
Okay. Okay. So another question for you guys. Uh, do you mean you use like trio sequencing files? I don't know what that means anyways. Trio sequencing files. Do you have an idea about that?
I most probably, I guess, using three files, three different data sets, like the
Okay.
maternal, paternal, and the offspring. I guess that is
Oh,
what
okay. Okay.
trio-sequencing means.
Okay.
We can use trio-sequencing. That is when we use a family data set. That is, we
Okay.
are looking into, let's say, inheritance of DMD from the mother to the offspring. That could be the case. Or else, if we can even use this exome sequencing on population data directly, like looking and comparing the genes or the expression of the gene with one population to another.
So what do you anticipate? Okay, there's another question, but I want to ask my question. So what do you anticipate from this study? Do you think DMDs will be more from the paternal or from the maternal side? Just
Yeah,
by intuition
sure.
from
It's gonna be from the maternal side because it's an X-linked inheritance gene, which is basically passing on from the mother to the offspring. And
okay.
this usually can happen, and the disease phenotype can be both dominant and recessive. That is, both the X genes for the female has to be both the carriers, or let's say the mother is carrier of the disease and the father is normal. So when
Okay.
the offspring, usually if it's a female, is going to be a carrier. That is, the disease phenotype is not expressed but is carrying one of the gene which can cause. But if the offspring is a male, basically he's going to express the disease. He's going to be one of the person who's going to express the disease, the DMD.
Okay, one more question. How do you plan to be inclusive in your data sets? Because I don't know if Duchenne muscular dystrophy, GMD, right, is, I don't know if there's any, human population, maybe African or Asian or Americans or Europeans, or, that it's more specific to. So do you plan to be globally inclusive or continent specific? So what's your population like?
So right now we did research on the data sets available for this process. So there were two kinds of data which were available. One is called the family trio sequences, that is passing on from the maternal paternal to the offspring. That is one kind of data set which we found. And the second kind, we weren't, it is not specified for the particular population. It directly states the, let's say a single person's data set is being compared with the reference genome of the entire of that particular chromosome. Let's say the chromosome 13 or 14 contains this DMD gene, uh, and this particular gene is going to be compared between the deceased person and the reference person to see what variant is going to be identified from the normal person, and is that particular variant is going to be expressing that particular phenotype. Because not all kind of variants and mutations have the similar effect as caused for DMD. That is our basic plan for using the data set.
Okay. Okay. So you have some other questions. How is exome sequencing different from whole genome sequencing? I think this is not a project specific question, anyways. But if you have an answer, you can answer that. I mean,
Yeah, sure. Yeah.
are there other people from your team on the call?
Yeah, we do have. Yeah, we have
Yeah,
Nigar and others.
yeah, they can also assist. It's a team
Yeah,
work.
sure. Nigar, do you want to answer the question?
Oh, sure. Okay, speaking of exome sequencing, we just use exons, the coding region of the sequence, but when we do the whole genome sequencing, we also have introns, the non-coding regions, and also the introns. So yeah, this is the difference, but I think this is what the question is meant to be. Also, we have another question too here.
Yeah. Go ahead. Apart from inheritance, what other factors can make individuals prone to DMD? That's a very good question. Apart
So
from
yeah,
inheritance, yeah. Go ahead.
yeah, there are a lot of reasons which can cause DMD, because DMD can also be caused by spontaneous mutations. It doesn't always follow the part of inheritance. Let's say there are various factors which can involve, such as, let's say, physical mutagens or chemical mutagens. such as UV rays, gamma rays, or certain chemicals which can directly induce mutations. These kind of agents can directly cause spontaneous mutations in that particular person. If the mutation is, is quite large enough that our DNA repair system can't repair that SNP or frameshift mutation, then that person is going to express the particular genotype of the mutation, which could result, let's say, in degradation of muscle, which in later onset in his life is going to worsen the condition if it's not treated, which could directly lead to DMD.
Okay, I think I saw Igerin, one of our very vibrant teaching assistants. Do you have a question, Igerin? Igerin, okay, I can't find that anymore. Sorry. Okay, I can't find that. Anyways, Michael, are you on the call? Do you have a question? I can see Michael in this.
No, I don't. I don't have one. And I don't have any questions for now.
Okay. Okay then. Next team. I think. Yeah, we have a question. Should they go ahead with the project? Everybody, I think they were able to defend their project clearly. For me, I think if you agree that they should go ahead, you can just type yes in the chat. And if you have if you have any objections, type no. Feel free to do that. Yeah, okay. The yes have it. We can go to genomics 2. Genomics
Sure,
2.
great, thank you.
Yeah. Ciao. Genomics 2. Anybody from Genomics 2 right now? Genomics 2, we're waiting for you guys.
Hello.
Yeah, please go ahead. Go ahead. Straight up. Introduce yourself and just.
Yeah, sure. Hello, everyone. My name is Romy Roy, and I'm from India. And our project is not so complex. It's fairly simple. It's based on malnourishment. We know one of the greatest gripping factors currently the world is facing is malnourishment. And the problem with that is the government nor the youth are quite confused whether it is the lack of food or whether it is the lack of nutritious food that is driving malnourishment. Everybody thought either these two are the reasons until a study came that suggested two groups having the same nutritious food and one group developed malnourishment. Now that changed the whole game. Now everybody is not confused. They are trying to find out, is there a genetic component for malnourishment? Not only genetics, is there a genetic component? Is there epigenetic factors? Or is there any variations or dysbiosis in our microbiome? And that's the question we are trying to address in our small internship. So our basic idea is to collect data, a WGS or an exome sequencing data. I mean, we are not sure where to get data, but I'm sure there is a database from the previous studies. Now, after that, we'll do a quality control and use FASTA QC, and we'll do the mapping and followed by calling of variants and annotating variants using SNPF. And we'll try to find the variants using data manipulation techniques. Now, that is for finding the data, the genetic variant. Now, we are also interested in knowing if there are any differential methylated region. For that, the same steps will be followed till mapping, and after that, they will do methylation bias using methyl decal and the visualization using plot profile, and we will detect the differential methylate region using methylene from the same Galaxy platform. So it's a very small project, but the implication is huge. Thank you.
Questions, guys? So type your questions in the Q&A, in the chat. My question is, I used to think, I mean, that's my belief, that malnutrition was from poor food regimen, that is bad nutritional regimen, not from any malnutrition does not look like a genetic disease, except you are telling us that malnutrition is there are some people that are naturally predisposed to malnutrition, which I might want to argue with you, or you are trying to know the genetic, or rather say epigenetic consequences of malnutrition on the genome. So that's what I want to know. I mean, I might, let me get your question, the question you are trying to answer. So are you trying to know the genetic variant that predispose a person to malnutrition, or you want to know the epigenetic implications of a malnourished of malnutrition on the genome? I mean, yeah.
So I think
Did you get
looking
my
at
question?
both
Did you
will
get
be
my question?
Yes, the first question is that you're confused whether it has a genetic variant or epigenetic component. That's a valid question, but we know, I mean, malnourishment in a way is caused by non-absorption of nutrients. So the non-absorption of nutrients, in a way, in our system, will be related to bioaccessibility and bioavailability. And bioavailability of nutrients, of course, will have a gene associated to it. So if there is a variant on that gene, it will I mean, everybody will have a different kind of absorption ratio. So we need to, our focus is on finding that genetic variant, or is there a genetic implication? Now, as we know, what is epigenetic? Epigenetic is nothing but, I mean, the accessibility towards the DNA sequence will be reduced by chromatin modulation. So if there is a genetic or a genetic variant, there is a high possibility that there will be also an epigenetic component to it. So what are the epigenetic component? We don't know.
Well, yeah, that's
it's an investigation.
I think I get your point now. So it's about possibility that malnutrition, probably a possibility or not, but malnutrition is influenced by relative absorption rates of individuals.
Yes.
Even in the presence of good feeding.
Yes.
Okay. Yeah, your question is quite interesting. So, Igeren, I think you're here. Do you have any question, any thoughts about the project? Michael, and anybody in the audience? Okay, someone is asking what type of data.
So we have started searching for the database. So as a preliminary search, we will use the data that we get our hands on for a proof of concept.
Okay. I wish you best of luck in that. But yeah, Igeren, okay, someone is saying it would be great if you could clarify if you are focusing on the digestive enzyme inactivation caused by sorry.
Yes. So digestive enzyme inactivation. So enzymes are nothing but proteins, right? So from where does protein comes from? Protein comes from mRNA. From where does mRNA comes from? mRNA comes from DNA. And how does DNA from mRNA comes out? It's due to the chromatin modulation. When the chromatin exposes the DNA get transcribed to mRNA. So if we break to the basics, that question is nothing but epigenetics.
Okay. Any more questions? If there are no more questions, I don't know if I should pass.
I think I have a question.
Okay, yeah, please go. And after now, please, Michael, I have a call. Please. Thank you.
Okay. Okay. So my question is based on the data that you want to use. Say you want to use an whole genome sequence data, right, from a population of malnourished people. So have you gotten his data set already?
No, we haven't got. We are looking for the data set. So there are some studies, which have used some data sets. So we'll just look into it whether we can use that as a proof of concept, not as a confirmatory thing, but as a proof of concept. So as I said, this is an
Okay.
investigation. We don't know what will happen. We'll only know once we perform all this stuff.
Okay. I think the pipeline is pretty straightforward for the variance calling you are doing. I think I can see the proposal. It looks straightforward. I hope
Yes.
turns out well.
Okay.
So does anybody have any other question? Wale is away from me. Do you have any other person in the audience that has any other question or question in the chat box? Okay, so if there's no any other question like we did for genomics one, I think we can vote for their proposal to be accepted. So you can type in the chat box. We want the proposal to be accepted like we did the other time. So just type in yes in the chat box. Okay, I think you have a lot of yes, so we can just move on to the next group, and that should be transcriptomics. So let's have the transcriptomics guys present their proposal. So anybody from transcriptomics can present. Someone from transcriptomics here. Anybody presenting for transcriptomics? Nobody. How about data science?
Okay, drug development is sorry. The guy said it's
Who's supposed to
partially
go?
slow, and
Okay.
Quadrant, can you go ahead?
Christian, are we?
Sorry,
Hello.
who is speaking?
Okay.
Who is speaking now? Sorry.
Yeah, I'm Christina from Transcriptomics. Are you asking for Transcriptomics group?
Okay, transcriptomics. Well,
Yeah.
okay, go ahead.
Okay. Yeah, I'm here on representation of the team Transcriptomics.
Can
So,
you quickly introduce yourself? I can't see your name. Sorry.
yeah, my name is Chris Rizu.
Oh, okay.
Okay. So I will tell you
I'm
about our
transcriptomics. Hello. Hello.
so, hello.
Please,
I don't know who
who is presenting for? Chris, please go ahead. I don't know. Somebody just came in just. Mute
Sorry,
yourself if you are not speaking.
I think maybe some sounds around here, but I'm not work. Sorry. Okay. I will tell you about our proposal.
Mhm.
It is still in an early stage, but it's passed in two studies about the COVID progress. One of them is a meta-analysis where they are studying the differences in transcriptomics, single-cell transcriptomics, in patients with COVID, depending on the severity of the illness. And the other one is studying also transcriptomics, but passing in the age of each patient. So since in these studies, we can observe differences between the cellular profile in transcriptomics in the different patients with different ages and also with different severity. So I would like to do a meta-analysis with the publicly available dataset to study these patients by two segregations, age and also severity, and see if these differences in transcriptomics is passed individually by these two factors, and we can also observe same differences between severity in the different ages. I mean, For example, the transcriptomic profile is more similar between young and old patients that have severe COVID, and not so similar in all the young patients, for example. And then we can use this to say, okay, this transcriptomic profile can serve as a biomarker of the severity of the illness. So this is our objective, and have to start to collect all the samples to start with the analysis, because I think that all of us are new with this single cell RNA sequencing approach, but we will be working in the R environment with the Seurat software as one of the papers that we have based on. So that's more or less all that we have till this moment. If you have any questions about, I'm happy to hear you.
Yeah, guys, do you have questions for the team? If you have any questions, you can unmute yourself and talk. Okay, for me, I wanted to ask, what do you expect to see, especially in terms of the expression of immune cells in the different age groups? What do you expect to see?
Yeah, I think that we will expect that the adaptive immune will be more represented in the patients younger, and also in the patients that has non-severe COVID, because it's related with the good prognosis of the illness. And we are expecting that in an increased innate immune response in all the severe COVID patients.
Okay. I think that's hopefully you get the results. I mean, sometimes we anticipate it. Yeah, but I just wanted to know something like your base and your expected results. I really don't have much questions. I mean, I've read the proposal, and it's quite straightforward and simple to understand. Not so complex. Okay. Does any other person have any question? And okay, maybe I have a question. I have a question, I think. But guys, do you have questions? Okay, I can't see any question. I would ask mine. Okay. Okay, so don't you think is there a correlation between age and your like your native immune response, like your normal immune response? Is there a correlation between that? Like normally outside of COVID-19, is there a correlation between age and? Hello?
Hello, can you repeat the question, please? Because I didn't hear you so well.
I said, is there a correlation between age and your immune system? Like, is there any relationship? Does the age affect the immune system?
Yeah. It's supposed that there is a correlation between age and the transcriptomic profile and the different cellular profiles. That is also observed in the COVID patients. Normally, you have more adaptive response than immune response when you are exposed to virus, and it's supposed that a bad response to the virus has a higher profile of immune response in the effect of the adaptive one.
Okay.
And
so
also,
okay, yeah, someone is asking a question.
No, I'm in the same group. I just want to contribute. So
Okay,
in normal immunology, we get that as one grows or age, and the immune system, develops. And so I think the older might have a better and more, let's say, more, maybe the aged may have a better immune system as compared to the younger ones, maybe the baby, or let's say the ones with the a very, very
Okay,
young
okay.
age.
Basically, what I understand is that I don't think it's a linear relationship. I think it's like when you are younger, you are kind of developing your immune system, and it's going up. Okay, but there's an age. I think around 60, 65, the immune system begins to decline again. I don't know if I'm right.
Hello, no, may I come in?
Yeah, yeah. Go
Hello.
ahead. Yeah, go ahead. Go ahead.
Okay, I'm also in the same team, transcriptomics. My name is Victor. Actually, immune system becomes compromised as you grow so advanced in age. And I also wanted to chip in that our research work will also help explain why the COVID-19 is not ravaging so seriously in Africa compared to other climates. It could be as a result of our response, immune response, maybe due to the things that we are exposed to in our environment. So that is one way we're also looking at in our research. So
Okay, well,
this is
that's an
going to
interesting
help us
question.
explain. Yes.
That's an interesting question I would like to pick up on. You said
Can I come in? It's not a question, it's a contribution. It's a contribution. I'm also a member of the
it's
team.
an
It's not a
interesting.
question. I'm just explaining the
For
basis
me,
of
I
the
see it
work.
as a question. So
Yes. So
Wale, can I come in?
yeah, sorry. Let me. You can come in, but let me ask the last speaker before I forget my question. So you were talking about Africa being stronger. I know what you mean. You are probably from Nigeria already. I'm also from Nigeria.
Of course. Yes.
I think the decline in testing is probably the most important reason for
that is
for the reduced prevalence in Africa, in African region.
Yeah, the decline in testing. The decline in testing is one factor, but also our response to it is another thing, because in Africa we have a lot of endemic into this kind of. So
okay, so
this
what
switch
is
from
the
innate to
pathogens that are
whatever will
endemic?
also help.
Yeah, okay, listen to me. The pathogens that are endemic in Africa, I'm not sure most of them are viruses. Yeah, there are a few viruses, of course, a few viruses. HIV is one of them. But I think
we have Ebola. We have Ebola.
But I think it's almost one already. Anyways.
We have Ebola too.
Okay. Anyways, the South American region also has a lot of viruses. Asian regions also have that. And if we cannot, you see, reporting public health surveillance, yeah, like Esther is saying, I can agree with you as well, is a very important key. Yeah, I mean, it's very important before you can be sure of some claims, okay? But then if you think it's discoverable, anyways, I didn't find that in your report, okay? I didn't find that in your report, but by the way, are you using, you're using patient immune profile. Okay, that's interesting. Someone wanted to come in the other time, sorry. We had a lot of arguments.
Oh yeah, I was the one. Wale.
Yeah, go ahead. Oh, Samson. Oh. Yeah, sorry. Where will we? I don't find. Where is this thing hiding people's name? I think I should go to the front.
So yes. I think sorry I joined late. But there was a part of the argument that caught my attention: immunity and age. And just to add to what you were saying, I think it's not a very linear kind of relationship when it comes to immunity and age.
Okay.
Because when you look at it, the baby, even from the womb, it's already expressing IgG.
Mm-hmm.
Are we together?
Mm-hmm.
You get, and then before it comes out, there's IgM already, and the rest of this. But that doesn't mean that even outside the body, the adults don't have this. I don't know whether you understand. It's always also present in them. So you can't say because they are baby, they don't have this. Though they have, it's still developing, but that doesn't mean that their body will not react the same way an adult would if they're exposed to certain antigens in the body. I don't know whether I'm clear.
Yeah, okay. By the way, for the record, Samson is a medical doctor in practice. So if he's talking, he's probably talking not just from the scientific perspective, he's also talking from the clinical perspective. They are part of the frontliners, and I don't want to be on that front line, actually. Okay, COVID-19 has raised enough problems for us, but I think you guys pretty understand your project. Yeah, it's. By the way, what was the conclusion for the last team, Michael?
Yeah, I think we passed them. Everybody voted them.
Okay. So, guys, can you vote for them? Do you have yes or no? Should they go ahead? Should they continue with this project? Personally, they can go. Yeah. Okay. Thumbs up, guys. Interesting speech. Interesting presentation. By all means, you guys should go ahead. Thanks to Chris for leading the team so far. I think I've seen you already. Yeah, and I was really happy when you came on board. By the way, is any so going forward, the next team is drug development. Guys, are you here? I hope your speaker is still.
Yes, we are here.
Okay, please go ahead.
Okay. Good day, everyone. My name is Omote Shokodri, and I'm representing drug development, presenting a project titled Screening and Molecular Docking of Plant Bioactive Compounds on Protein and Enzyme Precursors in Alzheimer's Disease. In Alzheimer's, Alzheimer's is common among the elderly, and then there is memory loss and loss of cognition. And there is a characteristic accumulation of the protein beta amyloid and the protein tau protein. Now, our project will be exploring two major precursor pathways to development of this protein, which is the amyloid precursor protein. We're looking for agents that will bind to the amyloid precursor protein, which produces beta amyloid protein. And then we are also looking for an agent that binds to the secretases that could enhance or inhibit the secretases that cleave amyloid protein from the precursor APP. Now, we are tapping into the safe profile and the potential of traditional medicinal plants. And then we are looking at after some exhaustive study of plants that have potential for Alzheimer's, we've identified 5 plants that we are going to be docking their metabolites on the proteins that are responsible in Alzheimer's disease. And then we'll be comparing them with current known plant metabolites that are known to improve the symptoms. And then we also be comparing them with some cholinesterase inhibitors that also improve symptoms too. We are looking into the potential of natural plants, and our pipeline starts from identifying the natural plants and the proteins we are downloading them from the protein data bank, and then we are also going to compile the database for our natural bioactive plant from MPAS and the Dr. Duke's ligand library for ligand Dr. Duke's library for ligands and natural plants. And we are going to do an active site analysis using the Discovery Studio and CASP, and then we'll label this active site using Pyrex before we then push our prepared products into Autodock Vina for docking. And after docking, we'll then sort them, and then we'll look for the ones that have high binding affinity, even compared to the current known treatments. We'll look for the ones with high binding affinity. We'll bring them out, and then we now screen them for ADMET qualities, looking at the absorption, looking at the drug, how the tendency of them to be developed into drugs. Looking at the toxicity, we use Swiss ADMET, and then we do Protox 2 for toxicity, check the toxicity, and then we look at then after we've seen this, we used to screen out some plant metabolites that has high binding activity that are not very safe in human, or they are not safe as drug options. And then we are also now, after that, we now go past our products into molecular dynamic simulation, and we'll simulate so that we can actually get the protein conformation, and then we know the binding interaction. We know the specific atoms and the proteins that are binding together in our ligands and our protein, just to provide more information on this, just to give more information to the natural metabolites that we have used, and to provide a potential for their use in drug development. Now, our expectation is to get a plant metabolite that have high binding activities, affinity for these major precursor proteins that have been identified, even more than the currently identified ones, thereby improving the current knowledge on plant use in Alzheimer's disease, and also creating potentials for development of new therapies for Alzheimer's. Thank you very much for listening.
Yeah. Questions, guys? Questions. Gentlemen of the press.
I don't have any question. Just that I like his presentation. It was nice. Even I don't have any question.
Okay. I have a question. I mean, I didn't see any targets. What are your targets? Your biological targets?
Exactly. What is the project trying to answer? What is really the key thing you want to solve about Alzheimer's using your plant, if you discover something? So
sorry, thank you very much for the question. Currently, there is no known cure for Alzheimer's, and there is large amount of research that is currently directed into developing new treatments for Alzheimer's, new and safe treatments for Alzheimer's. The current established is they are just memory enhancers. They improve the memory, reducing the progression of Alzheimer's, but there's currently no established therapy for Alzheimer's. So we are looking at contributing to that body of knowledge, and even significantly developing a plant metabolite that will be very active against this condition. That's the problem we are trying to solve. Thank you very much.
Yeah, so also answer my own question. What's your biological targets? I mean, your proteins. I know you said
Yes, our biological target. I said we'll be targeting two major precursor proteins in beta amyloid protein, in formation that are implicated in Alzheimer's disease. One of them is the amyloid precursor protein, 1-AAP, from protein data bank ID, and it is the protein that is cleaved by the beta and the gamma secretases. The protein where the beta and gamma secretases cleave the APP, the amyloid precursor protein, to form beta amyloid proteins that is implicated in Alzheimer's. So we are looking at binding.
So let me get a point. So IAPP is cleaved into APP.
Yes, by beta and gamma secretases.
Okay,
so
why
now
don't you target,
we are looking
why don't you target the enzyme that performs
the problem? The reason why we can't target the enzymes is because that means we'll be inhibiting the actions of the enzymes.
Yeah,
the enzymes are also involved they are also involved in some functional activities. They are necessary with other proteins in some functional activities in the brain. And then if we inhibit it, it affects that other activity. And that gives us our second, our enzyme target. We are looking at the alpha secretase, which is the order of, which also cleave the APP, but does not form beta amyloid protein. So we are looking for agents that bind to alpha secretase, so we can develop something that can enhance the activity of alpha secretase over beta and gamma secretase. So if we enhance the activity of alpha secretase over this, therefore there is reduced action of beta and gamma secretase in forming beta amyloid proteins, and therefore there is a reduction of the amount of beta amyloid protein that will be produced in the brain. So we are looking at those two.
Yeah, don't you think there will be? Now you are looking for something that will be specific for one and not specific for the other, right? If I get it.
We are looking for the ones that have high binding to one.
Yeah.
And would not display. Yes.
So
yes.
Are the secretases not structurally similar? Do you think?
They are structurally similar, and I think that is why we are looking down to their we are looking down to molecular dynamic simulation.
Yeah.
And also we are also considering the APP, the amyloid precursor protein. We are looking at the ones that would bind it and would not allow beta and secretase. So we are looking at the two pathways. Thank you.
Any other person in the team wants to contribute? Maybe we should give everybody a fair chance.
So sorry, I wanted to ask a question again.
Yeah, go ahead.
Because Alzheimer's is very interesting, and there are a couple of drugs that have been used, not since they are memory enhancers. I don't know whether you took your time to look at drugs like dompencil and their effect on acetylcholine. Can you just try and match them up? Let's see.
As we look at the effect of donepezil, tacrine, and we looked at increase and increasing the amount of the action of acetylcholinesterase inhibitor in ACH neurotransmitter in the brain can improve
acetylcholinesterase. Acetylcholinesterase.
No. ACH action, improving the action of ACH in the brain.
By inhibiting by inhibiting acetylcholinesterase.
Enhances memory. By inhibiting the cholinesterase inhibitor. Yes.
Okay.
Yes. We considered that in choosing our plants. Actually, we considered that because we also looked at plants that has this activity, having cholinergic activity too, and that is why we have identified these 5 plants. Yes.
So the plants. The plants you're looking at, they are more of they're cholinergic in their action, right?
They have we looked at the current research on drugs and the plant metabolite that are used in this condition, and then we looked at the plants. They have about 4 or 5 qualities. One of them is that they have cholinergic activities by improving cholinergic functions in the brain. Secondly, they are usually flavonoids. Thirdly, they have anti-inflammatory and antioxidant properties. So we combine these 3 knowledge, and then we use it to develop plants that have metabolites that exhibit these qualities. And we are also now going to look at how they can improve, they can bind to these precursor proteins and help this disease.
So yes, sorry. Maybe it's something that you could work on. Sorry. I'm taking time. Sorry. It's really interesting because I like what you're doing. And it looks like you have the stuff. So I want to involve you a little, and let's see 1 or 2 things. So cholinergic agonists, they are either reversible or irreversible. You get?
Yes.
So your 5 plants, are they going to be reversible or irreversible?
Well, we can't. The truth is, they are still bioactive plants. Even the molecular docking has never been done for these plants. So there are a lot of potentials for this our research, and
of course you can add it to what you want to discover.
Yes, definitely. It's something we'll note that down. Thank you very much. We would also
you're welcome.
Thank you.
Great work. Yeah.
Thank you.
Can I add 1 point?
Go ahead.
Hello. Yeah. So from what I understood, that you'll be focusing on specific plants, and from that plants you will screen out the metabolites, right?
Yes.
So that will be very limited in the sense, if you can perform a virtual screening at that step itself, you can get millions and millions of metabolites. You can do a virtual screening and you'll get the best candidates that's already in the gene bank.
I'm sorry, I'm not getting the question. I'm not getting the question. Sorry.
So currently, you are focusing on some plants.
Yes.
You're not so sure whether its metabolites will work. So instead of that, at that point, if you can perform a virtual screening, at that point, from that database, there will be millions of metabolites, right?
Yes.
So if you can perform a virtual screening at that point itself, you will get the best candidate metabolites at that stuff. So that metabolites might be from different sources, but it will be the best candidate, perhaps.
Okay. I've noted that down. Although we also considered that too. And then in our pipeline, we've already made arrangements for how we would assess the best metabolite for in our tests. So we have considered that. But thank you very much for the input. I would note that down. Thank you very much.
Okay, any more questions? I'm enjoying this. Okay, if there is no more question, I think we have one more team. I wish we had like 10 teams today.
Yeah, I think I like it.
We have data science, right?
Yeah, good evening.
Yep.
We're here.
Okay. Oh, yeah. Go ahead.
All right. So good evening, admins, teaching assistants,
One
and
second, James.
co-interns.
I'm really sorry to interrupt, but there is a process that you missed for the previous team. You'll have to ask whether the project should be going forward.
Yeah.
Oh,
Okay. They needed their verification. Okay. Guys, should we allow them? By me, I think they are super. It's a very super team already. They can go ahead, but someone said yes, no. Yeah, somebody said definitely yes. Okay, guys, data science, please go ahead.
So my name is James Uche. I'm representing data science team. Our project is titled Diabetes Prediction using machine learning in Python. Now, diabetes mellitus is a disorder in which the body is unable to normally regulate blood sugar levels. Hence, it results in high blood sugar levels. People who have been diagnosed with diabetes have higher risk of getting heart disease, kidney disease, stroke, eye problems, and nerve damage. And there are about 4 types, but our focus will be on type 2 and pre-diabetes. A 2019 study shows that about 463 million people suffer from diabetes. Approximately 4.2 people have died from diabetes per year. It is also said that by 2045, these numbers will rise to 629 million. Now, about 374 million people are at an increased risk of developing type 2 diabetes. In current medical diagnostic methods, there can be 3 different types of errors. The false negative type, in this case, the patient is already diabetic, but test results tell that the person doesn't have diabetes. And there's the false positive. In this type, the patient is not diabetic, but the test reports say the person has diabetes. The third type is unclassified. This means that the diagnostic system cannot diagnose a given case. This happens due to insufficient knowledge extraction from past data. And so a patient may get categorized in an unclassified type. However, in reality, the patient must be categorized either as diabetic or non-diabetic. Such errors in diagnosis may lead to unnecessary treatments or no treatments at all were required. In order to avoid and reduce such occurrences, there is a need to create a solution using machine learning algorithms and data mining techniques, which will produce high accuracy results and reduce human error. Our methods will be as follows. Well, since this project is a predictive analysis project, it will follow the supervised learning design using machine learning algorithms, and we'll start with data collection, data preparation, model selection, model training, model evaluation, parameter tuning, and prediction. We're optimistic that by the end, we would have created a tangible system that predicts with a high accuracy the likelihood of for a person to become diabetic based on their data. This will help clinicians make quick recommendations to help reduce the degeneration to full-blown diabetes. Thank you for listening.
Yeah, thank you for presenting to guys. You can roll out the guns and ask questions.
Yeah, I have a question.
Yeah, please.
Are there any variability in the parameters that are used in predicting diabetes, maybe diagnosing diabetes? Have you come across any data sets that maybe vary according to their parameters?
So I don't really get the question.
Yeah, so I want to ask whether the data sets, that you have, or maybe you've seen so far, there are variabilities in terms of the parameters that is used in diagnosing diabetes.
So you're asking if the parameters used to determine, or the parameters we assess diabetes, a diabetic condition or non-diabetic condition if they vary in different data sets, right?
Yes.
Okay, so we've only come across one particular data set now, but we intend to search for more, and then we'll probably look into doing those comparisons.
All right.
So I think there was a catch for me in your presentation. Nice work. And the catch was for clinicians to use, okay? And if clinicians are involved, that means you're working on human. You're already,
yes.
you're already using the work on human. So I wanted to know, because we have 4 ways we test for diabetes. We have the glycated hemoglobin, we have the fasting blood glucose, we have the random glucose, we have the 2 hours glucose test. So which of these tests do you guys want to use in training your algorithm in coming up with something that clinicians can use, you know?
Okay,
I think it's similar to my question. Yeah.
Yes, it's quite similar to your question. So from the data set we have, the parameters we have number of times pregnant, that's pregnancies, glucose levels, blood pressure, skin thickness, insulin, BMI, diabetes, pedigree function, age, and outcome. That's just from the data set. So, but like I responded to the previous question, we'll also search for more data sets and see if there are more parameters, especially in line with what you just mentioned, and see how we can work with that.
Yes, because it's something that we try to incorporate in normal clinical science now. You don't really use one of the tests to confirm for diabetes. You have to use maybe 1 or 2, okay, or 3 most times, you know, because you want repeated tests to be sure that what you are actually diagnosing for is diabetes. You get the gist, right?
Yeah, I do.
All right. Yeah, well done, guys.
Thank you very much.
I think I want to add to that as well. I'm also on the data science team, that like a machine learning algorithm can take in multiple parameters and even weight the different parameters. So if one glucose test has like a different or a lower sensitivity than another glucose test, then that can also be programmed into the algorithm so that like the variability or sensitivity is taken into account.
Okay.
So
I also have a question. Can I ask? Yeah. So my question is, you are testing for diabetes, like it's a binary question, either diabetic or not diabetic, right? I see. It's not quantitative in any way.
I think it's quantitative because the essence is not going to treat diabetes, right? It's going to learn the pattern and differentiate between people who have diabetes and people who don't have, and then it's going to be something that works like maybe let's use something basic like blood sugar levels. If it's getting to a certain point, oh, it's getting so high, you need to change some of your lifestyle patterns and all that. So it's going to take all those parameters and do the analysis and help make recommendations, especially about people's lifestyle.
Is it possible? Okay, yeah, go ahead.
Yeah, so when, as we mentioned before, this is the prediction models that we use are basically to reduce human error that we make. So when we are reducing human error, definitely we are increasing the effectiveness of any kind of treatment that we give, right? So one of the main important objective of our proposal is basically that.
Okay, so my other question was that, okay, yeah, but is it also possible that you guys can try to give us some other deductions about glycated hemoglobin, HbA1c? I think I can remember that. I once discussed that with someone. And it's a very important marker for diabetes these days. So is it possible you guys can, apart from just the blood sugar level, prediction of the blood sugar level or diabetic status, is it possible that you guys can predict the status of HbA1c as well? Glycated hemoglobin. Is it possible?
I think it ultimately depends on the data sets that we can get.
I surrender. Okay. Any other question, guys, before we pass them? Yes.
Oh, okay. Sorry, I'm coming back again. And I just wanted to understand something. Sorry. I think
it's an interesting discussion.
It's very interesting, you know. So I wanted to understand what you intend to do is to tell us whether someone is diabetic or not, right?
No, it's to compare data from diabetic people and non-diabetic people, and then it's going to be a diagnostic tool, more or less, where it does like it measures the likelihood or how close somebody is to becoming diabetic.
Okay, so it doesn't
Now it's a
mention
prediction
any of
model.
the pathophysiology, right?
Sorry, I didn't get that.
It doesn't get to indicate any of the pathophysiology. It's just to tell you this person is becoming is becoming close to having what's it called, diabetes, or is already having diabetes, right?
Yes, it just does if somebody's close to
okay, so diabetes. It doesn't mention why the person is close to it, right? That was what
like I said
it's
again,
just a
it depends. Yes, is making the predictions based on the data. Now, the parameters we use, right?
Okay.
If we use parameters that can link it back to the pathophysiology, then it's possible for us to also mention which pathophysiology may be causing disease.
Yeah, because if you look at the pathophysiology of diabetes, they are pretty much talk about so many things that could lead to increasing the sugar level in the blood. Just like Wale was saying, you maybe you need to find out one or two things about the glycated hemoglobin. It's very important diagnostic biomarker when it comes to diabetes. So I think you guys are doing well, and I like what you are doing.
Thank you very much.
Yeah, and kudos to Abdullah Kau on the chat. He has been backing you guys up on the chat box. Yeah, he has been. Okay, so do I have another question? I'm not sure. I'm trying to think about it. Okay, I think for now I don't have any other question. Yeah, okay, guys. Let's vote for them. Should they, from my end, I think they're a fantastic team already. But guys, it's up to you. Let's vote for them. Yes or no? Let's use the chat. Okay, nobody's voting for you guys. Okay, someone said yes. Okay, people say yes. Okay. Yeah, it's nice. I think I enjoyed my evening already. It's quite eventful, and I learned a lot as well. If you did not say anything throughout, can you just say hi? Yeah, it's okay. It's good. I heard your voice. Okay. So the next things will be delivered to you guys on Slack, but we want to give you this evening so that you can rest. We know just immediately after promotion, we give you another task. So till the rest of, I think we have about how many hours till to tomorrow? 6 hours approximately. I mean, if you're in Nigeria, anyways. So I'm also using Central European time, and we experienced daylight saving time, so we are back 1 hour. So I'm also using Nigerian time, and that was why the link got to you guys at exactly the right time. Like I was unawares before now. Anyways, thank you guys. It's been an impressive, wonderful session, and I think you guys have learned one or two things. So one thing again about your contributions, guys, your team leads are going to be responsible for your promotion this week. So I don't want to discuss, I don't want to tell you what the implications of that mean. If we assess you, so what you guys do is just to give us contributions, we try to assess that and try to have an idea based on our experience to know who contributed or did not contribute. But this time around, I think your team leads really know what's going on ground zero, and they will do. I think they will do good justice. And we don't want to receive any mails after that. Okay. But on the side of the team leads, try to be fair. Try to be fair. Of course, we share it. By the way, we took some pictures. I hope you guys will be kind enough to allow us to share it on our social media platforms. Yeah, okay, thank you. I saw you, John. So I thought John, is it John? I thought it's John Gemina.
It's Gemina. Gemina is my name.
So I thought you were a guy, one, and two, I thought you were from Nigeria. It's anyways, it's okay.
I'm from India.
Yeah, nice to meet all of you guys. Hopefully, by the end of this week, I think we should have another discussion. Watch out for Thanos and keep enjoying the learning experience. Any comments from Michael, Samson? I'm finding it difficult to communicate with you. Michael, Samson.
Yeah, I think I like this meeting. Yes, I think I like them, and we can have this more frequently, so that you shake up some persons a little, as they go through the process. Not only just sitting behind the phone and looking up data and the rest of it. There, you should be able to link the data together and come up with meaningful deductions, even in conversations, so that it's not just about you writing it down. But kudos to you guys. I'm sorry I came late, but you guys did perfectly. For those I listened to, you are doing amazing. You guys rock, and I can't wait to see you guys present something very wonderful by the end of the day.
Okay, guys. At this note, I think we should draw the curtain, right? See you guys. Let's continue tomorrow. You can go and rest for today. Bye.